Turns out the cancer immunology community is very cutting edge in terms of technology adoption, and they are among the pioneers to use CRISPR screens. And there are many CRISPR screens used in immune-related studies to answer many interesting questions. The first one is in vitro CRISPR screen, meaning that we grow the cancer cells and you treat with a cytokine, for example, interferon. If you remember, when the T cells detected cancer cell as being foreign, it secretes interferon cytokine. The cytokine will kind of kill the cancer cells, will also force the cancer cell to increase its antigen presentation to be examined by the T cells. And also very often when you treat the cytokines, the cell surface, well, they will also express the PD-L1, which is the molecule that makes T cell drunk. And so when we treat the cancer cell with the cytokines, as if we are bombing the cells with cancer cells with kind of like a bomb, and you ask, how does the cell respond? And in this nature study, they are looking at after the cells are treated with cytokine interferon, what gene influenced the cell surface expression of PD-L1 protein. So in this case, the cancer cells, the cell surface with interferon treatment will have PD-L1 expression, but depending on what gene is being knocked out, the scientists can sort the cells with those that have high PD-L1 expression versus a low PD-L1 expression, and use these two as treatment and control rather than day zero and the drug treatment. That you can use, after cytokine treatment with different gene knockout, which one has high PD-L1, which one has low PD-L1, and sort them into two groups and then compare their abundance of the guide. And using this approach, this is without interferon treatment, this is with interferon treatment. And the very top hit is PD-L1, which is not surprising. If you knock out the PD-L1 gene itself, the cell will not express PD-L1. It will go to the negative selection side. It can't really express a PD-L1. But from this, they identified another gene. It seemed to be important whether or not there's interferon treatment. CMTM6. This is a cell membrane protein which helps to stabilize PD-L1 expression on the cell surface. And so if we knock out CMTM6, then PD-L1, even though it can be expressed, it can be put on the cell surface, it's not going to be stable on the cell surface. And so this suggests that if you can somehow inhibit CMTM6, the cancer cell will no longer be able to stably express a PD-L1 to get the T cell drunk. And maybe the T cells can kill the cancer cells better, right? So that's an interesting novel target. And so this is using a cancer cell treated with a cytokine, and we're not looking at cell growth per se, we're looking at cell surface protein expression. In this case, a very important immune checkpoint that will inhibit T cell function, right? So that's one example. Another kind of CRISPR screen is a co-culture screen. This is, we have cancer cell with the CRISPR knocking out different genes, but we can co-culture it with T cells. And the T cells have T cell receptor to specifically recognize mutations in the cancer cells to kill the cancer cells. And we are asking, if the cancer cell knock out the different genes, would that make the T cell kill it better or worse? And so if you think about the original, in vitro first CRISPR screen, you just treat with cytokines, you use bombs. In this case, not only the airplane can have bombs, they can also shoot guns directly because remember, T cells not only can secrete interferon, it can use the fast ligands to induce cancer cell death. It can also use porphyrin to punch a hole and then shoot the granzyme into the cancer cell to make the cancer cell die. And so this is kind of, you now have the whole airplane, the T cell in there to do the killing, right? And so when we look at those genes, we see this is kind of the CRISPR screen result. By the way, in this experiment, they have two conditions. One is the T cells that really get activated, they contain a T cell receptor that really specifically recognize the cancer mutation. And those are the T cells that can just kill the cancer cell. The other is just general T cells. They may not have the correct T cell receptor to recognize the mutation, so they don't do the killing. And when we compare, this will be like treatment and this will be like control. We look at the differential abundance. You might see that some genes, if you knock out these genes, the cancer cell will no longer be killed by the T cells. And there are things like JAK-STAT pathway. These genes respond to the interferon in order for the cell death. And so if you stop the signaling, when the interferon bomb is coming, they just brush it off, they don't feel it. And so if you knock out these genes, the cancer cell will no longer responding to the interferon. They will be resistant to T cell killing. Whereas these genes, these are negatively selected, meaning that when you treat, when the T cell is there about to do the killing and you knock out these genes in the cancer cells, the T cells can actually kill the cancer cells even better. And there are different gene hits here. And the original study actually identified a protein complex, PBRM1, ARID2, and BRD7. These three genes form the same protein complex. And so it really kind of jumped out at the scientists. And turns out there was a companion study that showed that in kidney cancers, when the patient are treated with immunotherapy, this is kidney cancer treated with PD-1 or PD-L1, and if the patient has a loss of PBRM1 from mutations, when the PBRM1 is mutated, those patients actually will respond better to the PD-1 treatment. So you can see here, if it has the PBRM1 loss, those cancer cells are more sensitive to T cell killing, and they will respond better to PD-1 treatment, and the patient will live longer. But if they have the wild type PBRM1, well, they may or may not respond to T cell mediated killing, and so there you have a more cancer death. And so you can see here the CRISPR screens again here give us a immunotherapy response biomarker, and it discovered that if the patient has a loss of PBRM1, they will respond better to a PD-1 treatment. Another type of CRISPR screen is the in vivo CRISPR screen, where initially in the cancer cells, we infect them with CRISPR. Different cancer cells have different gene knocked out, and then we implant those cancer cells into the tumor. To let the mouse grow a tumor, you can also give the mice immunotherapy like PD-1 or PD-L1 antibodies, and you see how much that changed the tumor growth in the mice. So normally, in order to do a drug test on a mouse, every mouse will have to be given a different drug, and you see, or previously without CRISPR screen, you have to put the cell with one gene knockout into the mouse and see how much that tumor grow compared to without wild type cell, and you see is the tumor growing faster or slower. The nice thing about the CRISPR screen is in this one CRISPR screen, we can knock out hundreds of genes, and in one mice we implant hundreds of thousands or millions of cells in this one mouse tumor. You can evaluate whether knocking out of one gene will make the tumor grow faster or slower. So you can save a lot of animals in this case. So because in the mouse, the cancer cell has the CRISPR knockout, but the mouse not only have cytokines, it not only have T cells, it has B cells, macrophages, and like basically it's like the whole big immune system, right, with all the different weapons. And the Navy, the Air Force, and the armies are all together. And so there you really get a holistic view of what kind of genes in the cancer cell will make the cancer more susceptible to the immune tumor growth. Tumor growth under the immune system. And so using the CRISPR screen, this is kind of a work done by Nick Hanning's group. They found this drug target, PTPN2, this is a phosphatase, and they can really validate that in different models that normally the tumor will grow at this speed, but if you knock out the PTPN2, the tumor is growing much slower, right? So that kind of is a good example. You could even show that in this case, if so if the cell does not have immune system, that say the mice are nude mice, they don't have very good immune system, the tumor still grow fast, but with the immune system knocking out of PTPN2 really make the tumor shrink. And that would really support this PTPN2 as a very, very strong immune drug target, right? And I want to show you some other studies. So remember, this is the co-culture CRISPR screen of cancer cell co-cultured with T cells. In addition to PBRM1 or this protein, red protein complex, we also see this SERPINB9 gene. And looking at many other studies, this is in animals. In mouse, previously when scientists treated the mice with growing a mouse tumor and treated the mice with anti-CTLA4 antibody, and then compare the resistant tumor gene expression with the parental sensitive cell line. And they ask, what is the expression difference? They see that now the cancer cell or the resistant tumors are having higher expression of SERPINB9. We can also see this in multiple clinical trials. In those trials, if we compare the responders from the non-responders, so the responders are in red, the non-responders are in blue. We can also see that the non-responders have a higher expression level of SERPINB9. And so this is kind of from our study as well. We're asking, what is SERPINB9 really doing? So if you remember, when the T cells detected the cancer cell as foreign, it's using porphyrin to puncture a hole on the cancer cell and secretes the granzyme. It's like a poison into the cancer cells for the cancer cell to die. So T cells normally has these granzyme granules. These are bags and bags of granzyme in the T cells and NK cells. Anytime they get activated, they are ready for the weapons. They have bombs in some sense to be, or the guns to shoot into the cancer cells. The question is, these granules, they contain poison. Why don't they poison the T cells and NK cells? It's because SERPINB9 is a gene that can inhibit the granzyme function. And so they normally are in the T cells and NK cells to protect the T cells and NK cells being poisoned by the granzyme granule. Interestingly, because the cancer cell has genome instability, it's like, oh gosh, the granzyme is coming. So the cancer cell being unstable will also try to epigenetically try to express different genes, and it can learn over evolution that it can hijack a T cell mechanism and say, if I just overexpress SERPIN, then granzyme coming, no problem. It gets inhibited in the cancer cell. Then they wouldn't respond to cancer immunotherapy. And indeed, you see that when the cancer cells can upregulate their SERPINB9, the granzyme that's being sent from the T cells to the cancer cell will no longer induce the cancer cell for death. And that's also a very interesting immunotherapy response biomarker. And then I want to show you another one. This is also from a CRISPR co-culture screen. So by this time, there are many cancer cell co-culture screen with T cells. And another very strong hit is, so if we look at many different genes with glycosylation functions, so these can add sugars onto the cell surface or on protein surface or cell surface. And we actually use an algorithm to look at all the glycosylation enzymes. And the top one that the algorithm was able to come up with is this gene called MAN2A1, because in many, many CRISPR screens, we see that if we knock out the MAN2A1 gene, the T cells can kill the cancer cell more efficiently. We see this negative selection. We're like, oh, what happened there? And so we're able to indeed validate that if we knock out the MANTO-A1 gene, the T cells can kill the cancer cells more efficiently. And we use the MANTO-A1 knockout differential gene expression to generate a signature. And we ask, is it associated with immunotherapy response? It turns out the MANTO-A1 gene expression signature is associated with worse response in PD-L1 clinical trials. This one has 300 patients. And so in here, basically, when the overall glycosylation signals are very high, the patient has a worse survival. But if the patient have a low glycosylation levels, they have good survival. Oh, so this gave us some hints that maybe we can inhibit this glycosylation and the cancer cell will be more sensitive to anti-PD-L1 treatment. And so we experimentally tested this. There is a MANTO-A1 inhibitor. This is a small molecule that can inhibit MANTO-A1. And we tried this in both the melanoma mouse model, tumor model, and also the lung cancer mouse model. You can see here, even though the inhibitor itself or the PD-L1 itself doesn't really have a very strong effect, this combination is able to control the tumor growth much, much better, also prolong the mouse survival. And this suggests, again, from the CRISPR screens, not only we can see the resistance mechanism, we can identify novel, we can identify response biomarkers, we can also identify novel targets which can be combined with existing immunotherapy to improve the overall immunotherapy response. And finally, recently people are using CRISPR screens in the T cells. So previously we were using CRISPR in the cancer cells, but this particular study is using CRISPR in the T cells. You collect the primary T cells, you infect the cell, and in the different T cells, you're knocking out a different gene, and then they really profile the phenotype, such as T cell activation or T cell proliferation, making clones of itself, or T cell dysfunction. When does the T cell start to not do so well? And so using this approach, they can identify hits. Some of them are involved in T cell proliferation, others are involved in T cell activation, and also things that are related to T cell killing of cancer cells or T cell having dysfunctional state. And interestingly, after this study, they identified 50, 100 hits, and then they make a very small pool of those guide RNA, just targeting the hits, and then infect the T cells, and then they can do perturb-seq, which is single cell RNA-seq, to say, when you knock out those genes transcriptionally, what is happening to the T cells with those gene knockouts? And they can look at that as well for transcriptional signatures. So these are just some applications of CRISPR screens. So I hope that you can see that CRISPR screens can identify gene functions, which gene is promoting cell growth, which gene is inhibiting cell growth. You can find cancer vulnerabilities. You can ask, in this particular cancer cell line, what gene is gonna make the cancer cell die compared to, of course, you want the specific genes. You don't want pan-lethal genes, right? But you can see, okay, if the cell has a mutation in this gene, they will be vulnerable to another gene. That's, will be really, really cool to learn. You can use it to identify genes that regulate protein expression on the cell surface. For example, CMTM6 regulating PD-L1 expression on the cell surface. You can use it to look at the drug mechanism of action. You ask, what kind of gene knockout will make the cell more sensitive or less sensitive to a BRAF inhibitor? And this kind of give you a response biomarker as well. You can see drug resistance mechanisms, right? In this case, we see that if you knock out CDK6, or if you have a CDK4/6 inhibitor, it will synergize with the drug treatment. But then other genes, when you knock out, it will make the cell resistant to the drug. We can use CRISPR screens to find a lot of regulators of tumor immune response or immune cell function. And we can also identify novel drug combinations, whether it's targeted therapy or immunotherapy, to overcome drug resistance. And also we can combine CRISPR screens with other high throughput assays such as Perturb-seq to even understand more things about tumor immune or gene function, drug response. And of course, all of this, especially CRISPR screens, are generating a lot of data. And so the computational analysis and integrations are very, very important to really make sense from the data. And that's where we are hoping to get more students like you to be interested in looking at CRISPR screens. Okay, so that's all for today.